Okay, I'm going to give you the solutions for the two exercises. The first one, they gave you a strand with some nucleotides and with some nitrogenous bases, and you had to do the complementary strand. It's easy. If you take into account this, if you know this is just a game, because you have adenine, the complementary, what is going to be thymine, okay, so you put it here. If you have guanina, what you're going to have is cytosine underneath. If you have cytosine, it has to be guanine. Thymine, adenine, guanine, cytosine, it's always the same, it never changes. So this would be easy to do. I hope that none of you have had any problems, you will tell me. And the other one, the exercise 7, was about the differences between DNA and RNA. Okay, so we had to use this sugar basis, strands, location and function. There was another one, like a spatial arrangement, and I didn't understand very well what was this about. I understood that was location. I don't know. If you have, if you know what is it, you tell me, please. So the sugar that we are going to use in DNA is going to be deoxyribose, right? Desoxyribosa. That's why the D of DNA. The bases that we are going to have is adenine, thymine, cytosine, and guanine. Strands, two of them. It's a double strand. Location inside the nucleus, and I put here, if you want to add it in some organelles, you can have DNA too. It's in mitochondria and chloroplast. You can have some DNA too, but with nucleus it would be enough. And the function, the two main functions of two main functions of the DNA. First is to keep all the information of the individual, then do the replication for the regeneration of a tissue or the cell division or sexual reproduction. And another one is that they carry, it carries the information to produce proteins, right? So this would be the function of DNA. Then you have RNA. As it is a ribonucleic acid, the sugar is going to be the ribose, right? Then the bases are going to be almost the same except for the thymine. Here we don't have thymine, we have uracil, right? How many strands? 1. The location, you can find RNA inside the nucleus, the RNA, the messenger RNA or the ribosomal RNA. They are going to be in the nucleus, then they are going to get outside, and the function is, it depends on the type of RNA that we are talking about. If we are talking about a ribosomal RNA, to produce ribosomes. If we are talking about a messenger RNA, is to carry information from the DNA to outside. And the transfer RNA, the main importance is because it's going to be involved in translation, which is the production of proteins, and it's going to carry the amino acids. This is amino acid. The amino acids that are going to form my protein. Okay? I hope it was not very difficult. If you have any questions, you can tell me. Okay? Bye.